Hello, I am Hannah Fott, and I conducted research on the bioremediation of PCBs by native bacteria and fungi. The focus of this project is to observe the difference in consortium and diversity of native microorganisms between a large outdoor tub of PCB contaminated soil versus a small inlet pan of soil, if aeration and nitrogen promote bioremediation, and if there are genes in bacteria and fungi that possess PCB bioremediation capabilities. I predict that similar consortiums will be observed between pans. The large outside tub will have a greater diversity than a small indoor pan. Aeration and nitrogen will promote bioremediation, and that genes possessing bioremediation qualities will be identified. Polychlorinated biphenyls, or PCBs, are man-made chemicals that are persistent and ubiquitous in nature. PCBs are a known carcinogen and contaminate soil and water in Spokane, Washington at high levels. My research could be applied to using native bacteria and fungi to clean up PCB contaminated soil sites, which is both a natural and cost-effective process. A tub with 10 gallons of PCB contaminated soil was held outside. A small pan with 600 grams of soil was held in lab. Nitrogen fertilizer was added to both samples at a 40:1 carbon to nitrogen ratio and applied twice. Another small pan of soil was stored in lab as a control. Soil was aerated daily. Microorganism species were identified via 16S nanopore sequencing monthly. PCB and hydrocarbon levels were determined monthly using gas chromatography at Europe's Test America Spokane. In 2021, PCBs did not bioremediate. Diesel range organics were not detected, and residual range organics degraded marginally. Based on Shannon's and Simpson's indices, diversity decreased with little bioremediation. No consistent consortium increases for either non-degraders or potential PCB degrading bacteria during the study. A BLAST database was created from the assembled genomes of microorganisms in September 2021. Conserved domains of known PCB degrading enzymes were queried against this database to determine if they were present. Due to low E values, it appears that none of these genes were present. During 2020, contaminated soil was held in 2 lab pans. B received nitrogen and C received nitrogen previously bioremediated soil inoculant. In this study, initial PCB levels were 2,000 parts per million. PCBs were significantly bioremediated. Diversity increased, degrading bacteria abundance increased while non-degraders stayed consistent, and hydrogenase, a PCB degrading enzyme, appeared to be present. In 2021, bacterial and fungal consortiums were similar in the outside tub and indoor pan, so I have failed to reject the hypothesis that consortiums would be similar. It does not appear that aeration and nitrogen promote bioremediation PCBs, so based on this study alone, we reject the hypothesis that aeration and nitrogen promote bioremediation. It is unknown if genes with bioremediation abilities were present in the soil in 2021, so reject the hypothesis that genes would be present. Bacterial and fungal diversity did not differ between the outside tub and the small indoor pan, so I reject the hypothesis that the outside tub would have a greater diversity than the inside pan. Based on the study alone, this bioremediation technique does not work as hypothesized, but based on previous studies such as 2020, we believe that our conclusions are invalid. We believe that because initial PCB levels were extremely low, bioremediation did not occur. In previous studies, when initial PCB levels were high, bioremediation occurred and species consortiums and diversity increased over this process. In this study, consortiums did not change and diversity decreased over time, leading us to believe that with high levels of PCBs, the addition of nitrogen and aeration, native bacteria and fungi will bioremediate PCBs efficiently. Thank you.